Hi, in this video I'll summarize how DNA replication works. So DNA replication occurs whenever a cell has to divide. So that takes place, for example, during interphase before mitosis, as well as during interphase before meiosis. The process starts when the enzyme helicase breaks the hydrogen bonds between the parent strands. They're shown in dark blue here. So by breaking the hydrogen bonds, helicase separates those complementary parent strands and it untwists or unwinds the double helix. Then single strand binding proteins will stabilize the two strands, keep them separate by preventing hydrogen bonding between the complementary bases of the parent strands. Then each parent strand, the two parent strands shown in dark blue here, will act as a template to make a new complementary DNA strand. Primase is the first enzyme that will start to synthesize nucleic acids. So primase creates a primer, which is actually RNA. It's not DNA, it's RNA. And primase will synthesize a primer on each strand. So essentially it reads the parent strand and synthesizes an RNA primer as a new complementary daughter strand. Then the real process of DNA replication begins with DNA polymerase III, which will read the parent strands, actually both strands, both of the parent strands. It reads those parent strands 3' towards 5'. And what it does is it adds new nucleotides to the 3' end of the growing strand. Now remember the anti-parallel conformation of DNA. If the DNA polymerase III reads or moves on the parent strand 3' to 5', then it will make a new complementary strand in the anti-parallel direction, which means 5' towards 3'. Then DNA polymerase I will replace the primers. Remember, that's made of RNA. It will replace that with DNA nucleotides. That's important, otherwise you would be left with a whole bunch of RNA primers inside your DNA. And at the end of the process, the enzyme DNA ligase will form phosphodiester bonds between what used to be the primers, and that was just replaced with DNA nucleotides by DNA polymerase I, and the rest of the daughter strands. So DNA ligase essentially ligates. It attaches the bits and pieces of the new daughter strands that have to be attached by phosphodiester bonds. Now, you will notice from the graph that the two parent strands are copied differently. The leading strand, shown at the top here, is a little faster. That's why it's called the leading strand. You have to remember that enzymes such as primase, DNA polymerase III, DNA polymerase I, they move along the parent strand 3' towards 5'. So for the leading strand shown on top here, DNA polymerase and other enzymes move in the same direction as helicase. On the bottom strand, that one here is called the lagging strand, the enzymes like primase and DNA polymerase still move 3 to 5, but in this case, that happens to be in the direction opposite to that of helicase. So in this case, helicase moves to the left. It unwinds or separates the parent strands towards the left. So the replication fork moves left, but the enzymes can only move to the right. So what's going to happen then is that primase and DNA polymerase 1 and 3 will make bits and pieces of primer and DNA moving to the right, and then these enzymes will have to go back towards the left, towards the opening replication fork to create new fragments of DNA and RNA. So these fragments are called Okazaki fragments, and obviously they're going to be connected at the end by DNA ligase. So at the end of the whole process, the entire original DNA molecule is copied. So you get two identical copies of DNA. And it's important to realize that the process is semi-conservative, which means that you keep half of the molecules. Semi means half, conservative means keeping. So each newly formed DNA molecule has one parent strand, so the dark blue strand here, so that's one of the old strands, as well as one new daughter strand.